Real Science Review: Antibiotic Resistance. James, a future Farmers of America student, asked his FFA teacher, "Mr. Mack, why does the government say that ranchers should not use antibiotics in cattle feed? I heard somewhere that antibiotics actually make cattle grow better, which is why ranchers want to use them in the cattle feed." Teacher McPherson: "The government does more than just recommend. They actually test cattle at slaughterhouses and reject them if antibiotics had been used. The reason is that bacteria can become resistant to antibiotics. A few bugs will survive because they are naturally resistant. They create a whole new population that is resistant. All descendants now have this defense against the antibiotic. A new strain of resistant bacteria evolves rapidly, on a time scale of a few months or years. As more and more bacteria become resistant, we find that many of our antibiotics are no longer effective to treat disease. Many of these now resistant bacteria populations can infect animals as well as humans." Darwin's theory of evolution helps explain how bacterial strains evolve to acquire resistance to antibiotics. Nature favors or selects organisms that possess unique adaptations that allow them to survive in an unsuitable or toxic environment. Some bacteria have developed genes that confer resistance to antibiotics. As antibiotic use has increased in both animals and humans, more resistant bacteria are developing. Vocabulary used in the original research report: ACRAB TOLC multi-drug efflux pump, a gene sequence that codes for the ability of certain bacteria to affect certain drugs. See efflux pump below. Bacterial conjugation. Bacterial conjugation is the transfer of genetic material between bacterial cells by direct cell-to-cell contact or by a bridge-like connection between two cells. Drug resistance. Ability of an organism, such as bacteria, to resist a drug that normally would damage or kill the organism. Efflux pump, a biochemical system that operates like a pump to remove molecules out of a bacterium. Bacteria that pump out antibiotics may be resistant to antibiotics because the intracellular concentration of drug is too low to be effective. Gene expression. Genes are not necessarily turned on to perform their function. Those that are turned on to be active are called expressed. Gram-negative bacteria. The common dye for staining bacteria is cresol violet. A wide range of bacterial strains do not take up the stain, and these are called gram-negative. Horizontal gene transfer. Passing or transferring genetic information sideways to a relatively unrelated organism, as opposed to a direct descendant. MIC, abbreviation for minimum inhibitory concentration. It means the lowest concentration of an antimicrobial that will inhibit the growth of a microorganism after overnight incubation. MDR, abbreviation for multi-drug resistance or multi-drug resistance. In this research report, it refers to bacteria that are resistant to many different kinds of antibiotics. Plasmid, a segment of DNA independent of the chromosomes and capable of duplication, used in recombinant DNA procedures to transfer genetic material from one cell to another. Tetracycline, an antibiotic that is effective against a wide range of bacterial types. The drug is commonly used in livestock feed and in animal and human medicine. Tetracycline-resistant bacteria may infect animals or humans. TET-A, name for the tetracycline efflux pump. Efflux pumps are involved in the defense of gram-negative bacteria against two new antibiotics. Abstract. We evaluated the antibiotic activity of two naturally occurring new antibiotics, isopavaclone and diospyrin, on normal and multi-drug resistant, MDR, gram-negative bacterial cultures. The two new antibiotics exhibited antibacterial activity against several gram-negative bacteria in their activities. The antibacterial effect increased when we added an efflux pump inhibitor to the bacterial culture. Also, the pump inhibitor even increased the antibiotic effect in bacterial strains in which the pump gene sequence was incomplete. The overall results indicate that the new antibiotics could be candidates for the development of new drugs against MDR strains. Combining them with efflux pump inhibitors could reinforce their activity. Introduction. Antibiotics are drugs that kill bacteria, but not viruses. When exposed to antibiotic drugs, bacteria that escape destruction may evolve resistant strains. Drug-resistant bacteria can become dominant, and the infections they cause may no longer respond to antibiotic treatments. The problem is worldwide in both animals and humans. There is growing challenge in healthcare to find ways to combat resistant organisms. Approaches to avoid development of drug resistance includes improving control of early infections, using antibiotics appropriately, preventing the spread of strains that do become antibiotic resistant, and development of new antibiotics. One reason for antibiotic resistance is that bacteria have biochemical transport systems that eject antibiotics. These systems are called efflux pumps. If the genes that make these pumps are fully expressed, they make it easier for the bacteria to survive antibiotics. Efflux pumps have been identified in gram-negative bacteria and enterobacterial stains. Several chemicals can inhibit these efflux pump mechanisms and restore bacterial killing levels of antibiotics. A gene sequence called ACR-AB-TOLC helps to create efflux pumps. Thus, any chemical that inhibits expression of this gene would disable the pump and allow antibiotic to become effective. We tested here the ability of various bacteria strains to develop resistance to two new antibiotics or ability to develop resistance to the two new antibiotics, tetracycline and various other antibiotics. We also wanted to know if resistance they develop could involve efflux pumping mechanisms. Methods. Chemicals for antimicrobial assays. The new antibiotics were obtained from the Chemical Stock Bank of the Laboratory of Organic Chemistry, University of Yaoundé, Cameroon. As a basis for comparison, we used several established antibiotics: chloramphenicol and norfloxacin, tetracycline hydrochloride, imipenem cilastatin, and cefepime. We also tested iodinotetrazolium chloride, phenylene, arginine, naphthylamide, and triphenyltetrazolium chloride. Bacterial strains and culture media. The microbial species used included antibiotic-resistant and reference strains of Escherichia coli, Enterobacter aerogenes, Enterobacter cloacae, Pseudomonas aeruginosa, and Klebsiella pneumoniae. E. cloacae strains EC0769 and EC1194 were from the laboratory collection. Strains and their features are shown in a table that is omitted here. Prior to any assay, all strains were pre-cultured overnight on Mueller-Hinton. Mueller-Hinton broth was used as the liquid culture medium for antibiotic susceptibility tests. Bacterial susceptibility determinations. The drug and compounds were tested in the absence or in the presence of the efflux inhibitor PABN at a final concentration of 20 micrograms per milliliter. We determined minimum effect concentrations of the new and comparison antibiotics with a colorimetric assay. Briefly, the test sample and selected antibiotics were first dissolved in dimethyl and sulfoxide. The solution obtained was serially diluted twofold in a 96-well microplate. We then added 100 microliters of bacterial inoculum, prepared in MHB. The plates were covered with a sterile plate sealer and then agitated with a shaker to mix the contents of the wells. Plates were then incubated at 37 degrees Celsius for 18 hours. Wells containing MHB, 100 microliters of inoculum, and DMSO at a final concentration of 2.5% served as the negative control. A dye that would indicate surviving bacteria was added, iodonitrotetrazolium chloride, 0.2 milligrams per milliliter. These samples were incubated at 37 degrees Celsius for 18 hours. Surviving bacteria were indicated by a pink color from the dye. The MIC of the antibiotics was defined at the lowest sample concentration that prevented this color change. The samples were tested alone and in the presence of an efflux pump inhibitor, phenylalanine, arginine, nimbufilide, 20 micrograms per milliliter as described previously. We independently repeated each assay 3 times. Results and discussion. When used by itself, the pump inhibitor had no effect on the bacteria. Antibiotic activities of the 2 new antibiotics. We tested various normal and antibiotic-resistant bacterial strains for their susceptibilities to the 2 new antibiotics and to reference antibiotics. The 2 new antibiotics were more effective on some bacterial strains than the 2 common antibiotics. Isobavacone is non-toxic to healthy karyotic cells, suggesting that it might have clinical use. We compared antibiotic effect with and without the antibiotics in the presence of the efflux inhibitor, PABN. Table 1 compares the effectiveness against all strains of the 2 new antibiotics and the 2 commonly used ones. Interestingly, the activities of the 2 new antibiotics used against resistant MDR strains were better used than those of the commonly used antibiotics. Table 1. MICs of the various antibiotics on normal and antibiotic-resistant bacterial strains. The drugs and compounds were tested in the absence, negative, or in the presence, positive, of the efflux inhibitor PABN. Role of efflux pumps inhibitor. The efflux pump inhibitor PABN significantly increased the antibacterial activities of the 2 new antibiotics, with all MICs decreasing to below 10 micrograms per milliliter for the E. coli, K. pneumoniae, and E. coli strains. In addition, this enhanced activity was observed against various strains of E. coli, E. aerugines, K. pneumoniae, P. aeruginosa, and E. cloacae. All of the E. aerugines strains except 298 were resistant to both new and common antibiotics. Even so, the pump inhibitor made them susceptible to the new antibiotics. The pump inhibitor even increased the antibiotic susceptibilities of strains that lacked a complete pump gene sequence. This suggests that there may be an additional undiscovered pump mechanism that the inhibitor also inhibits. The efflux mechanisms clearly appear to be the first line of bacterial defense against antibiotics, as has been demonstrated for other natural compounds. This study provides evidence that efflux pump inhibition makes antibiotics effective against antibiotic MDR strains. Both strain KP55 and strain KP63 were reported to be resistant to most of the commonly used antibiotics, showing high levels of resistance to ampicillin, ceftazine, and azithromycin. MIC values up to 512 micrograms per milliliter. Here, we observed that all those antibiotic-resistant bacteria were susceptible to the 2 new compounds studied, especially in the presence of the efflux pump inhibitor. The study also demonstrates that the efflux pump mechanism is one of the primary active defense mechanisms of bacterial cells against these molecules. This indicates that medical treatment with antibiotics might be approved if given jointly with efflux pump inhibitors.